Well, hi, you guys. Thank you for joining us for this month's ICRE Expert Series talk. Today, I have the pleasure of having Kai Kopecky, who is a six-year PhD student candidate at UC Santa Barbara and works at the Moorea Coral Reef Long-Term Ecological Research Program. There, he combines field experimentation, underwater photogrammetry and mathematical models to understand how different types of disturbance events affects the ability of coral reefs to recover now and in the future. He received a bachelor's in science in ecology and evolutionary biology from UC Santa Cruz, where he worked in a variety of different ecosystems, including kelp forests, seagrasses, and tropical rocky reefs. Overall, he is interested in using scientific approaches to come up with creative strategy for ecosystem restorations. So with that, I will leave it to Kai.
Cool. Thank you. Yeah, thanks for the invite to do this. This it seems like a really cool program. So I'm happy to talk to you all about some of the research that I do. So I'll be talking about sort of what I've been working on in my PhD and relating it a little bit towards how I hope the stuff that I'm researching now can be used for management, conservation, and hopefully restoration of coral reefs. And then also how we might think about the similar ideas in other ecosystems as well. All right. Can you all see that screen? All right.
Yes.
Cool. All right. So you read or heard the title of my talk. I'm going to be talking about these things called material legacies and how they affect the resilience of ecosystems and how we might think about these material legacies when doing ecosystem restoration. And I'll mostly be talking about specifically these material legacies, which I'll explain what those are, and how they relate to the recovery of coral reefs. So, but before we jump into coral reefs, I want to just kind of set the stage ecologically for the kind of work that I do and like what I think about. So really what I'm interested in is what we call foundation species. And these are organisms that create the structure of an ecosystem and then provide habitat for other organisms in the ecosystem. And these are things like trees and forests, or oysters on oyster reefs, corals on coral reefs, or grasses and bushes and things like that, and maybe like a chaparral environment. So these are the organisms that basically make the structure and habitat for everything else to exist within. So I'm really interested in these types of organisms, but specifically what I'm interested in is what happens to them when they die? Like what do their sort of dead remains do in terms of like how they affect the recovery of the ecosystem afterwards? So there are varieties of different types of things that can cause these organisms to die. These are trees that have died from a drought, or oysters that have died from what we call a hypoxic event, where there's not enough oxygen in the water. Corals can die from a lot of different things, including coral bleaching, which maybe you've heard of, and grasses sort of just die every year seasonally. Anyway, what we refer to these sort of dead structures as is material legacies. It's some sort of material that kind of remains in the environment after something like a disturbance that kills them, and then actually affects the way in which the ecosystem recovers after that disturbance event. So while I'm interested in how these material legacies affect ecosystem recovery in general, the majority of the work I've done has focused on coral reefs. And so coral reefs experience all these different kinds of disturbances that can cause corals to die. Across this top row here, we have tropical storms like hurricanes or cyclones. They create really big, powerful storm waves that can kind of come through and break corals up and kill them on the reef. Things like when ships run aground or run ashore and actually just scrape corals right off the reef. Or things like dynamite fishing, where people actually throw explosives into the water to kill fish, but in doing so, they actually kill corals as well. And so in these cases, material legacies tend to be removed from the reef. So usually you don't have any sort of dead skeleton left over after these kinds of disturbances. On the other hand, these other types of events on the bottom row, like coral bleaching, where the water gets too hot and it actually causes the corals to get stressed out and die, or outbreaks of coral predators, like these crown of thorns sea stars in the middle here, that extrude their stomachs over the coral and then digest the soft tissues of the coral, or disease events like this black band disease that's killing this brain coral here. These are all things that kill corals, the soft tissue of the corals, but they actually leave the skeleton of the coral on the reef. And so in that case, the material legacy is retained in the environment. So I'm really interested in the difference between these two types of disturbance events and how they affect recovery. Also, by the way, if I say anything that doesn't make sense to somebody or use a term that you're not familiar with, you can feel free to stop me at any point, ask a question, put a message in the chat, or just, turn off your mute and call out. I'm totally fine with that. So I'm interested in how recovery is affected when material legacies are removed from a coral reef or when they're left on a coral reef. So I'll sort of show you what this looks like just with a couple of photos here. So imagine a typical healthy coral reef. This is sort of what it looks like, where you have all these healthy living corals kind of covering the entire reef. And they're providing habitat for things like this Moorish idolfish here, or this parrotfish over here. And you can see there's like a lot of complexity, a lot of little nooks and crannies for things to hide in. So there's lots and lots of habitat that these corals provide for other organisms. But when you have a disturbance like a tropical storm, again, like a hurricane or something like that, that creates these really big, powerful storm waves, they're actually so powerful that they can come through and break corals off the reef, even though they have these rocky skeletons, and the reef will sort of be left looking something like this, where it's sort of almost flattened. So then on the other hand, if you have something like a coral bleaching event, which maybe you've heard of a little bit, you have a really different outcome. All these white corals you see here have been bleached. So they got too hot because the water was too warm, and then they sort of stressed out and died. And it's a little more complicated than that. I'm happy to explain that a little bit later on at the end of the talk if you want to hear more about how that works. But anyways, all those dead coral skeletons sort of just get left on the reef, and it makes a really different habitat for the recovery to take place in. So then this really clear, obvious question comes up. How do the dead coral skeletons actually affect the ability of coral to recover after these different disturbance events? Is it faster when the skeletons are taken off the reef? Is it faster when they're left on the reef? And do you get different organisms coming back to the reef afterwards when you have skeletons versus when you don't? And we actually have had both these different types of disturbances happen in the place that I work. And we've started to see sort of some interesting patterns starting to happen. So about 10 years ago, or maybe a little more than that, on the island that I work on called Moorea in French Polynesia, there was a tropical storm, a cyclone, where it basically looked like that photo on the left there. And if you zoom in a little bit, you can see this just basically bare rock that's left there. This is actually a really good habitat for new corals to come in and settle onto, and then grow into adult corals and sort of allow the reef to recover. On the other hand, more recently, there was a big coral bleaching event in Moorea. And a couple years after that, we started to see something really different happen. So this is sort of what the reef looked like afterwards. There are a few live corals still you can see here and here. But we actually started seeing a lot of algae starting to like grow on those dead coral skeletons. And I don't know if you know about what algae does in a coral reef, but they actually are really bad for corals. They compete with corals for space and for light, and they can kill corals and to kind of overtake the reef. So we were starting to see this really different pattern taking place in terms of how the reef was able to recover. After the tropical storm, the reef recovered really well. But after this coral bleaching event, we were seeing that the reef was not really recovering as well as we would have expected. So I wanted to kind of look into what do the dead skeletons actually do to live corals and other organisms on the reef. And so I kind of mentioned this place already, but this is Moorea in French Polynesia. It's a small island. It's right next to Tahiti, sort of like, if you think about where Hawaii is, it's directly south of Hawaii, but on the other side of the equator. So maybe 1,000 miles south of Hawaii. And it's about 3,000 miles away from anything else. So it's really out in the middle of nowhere, out in the middle of the ocean. But it's a really beautiful place to work. As you can see, you have this like really clear blue water, some nice shallow reefs in the lagoon here, and then deeper reefs offshore here. And right here is a place called Gump Research Station. This is where I spend about 3 months a year. I just got back, about 6 weeks ago. And it's a really awesome lifestyle. You kind of like get to go live out there on this tropical island, wake up in the morning, you kind of walk down to the dock, you get your scuba gear together, your research equipment, you throw it in the boat, you hop in the boat, you zip out to wherever your experiment is, your field site. I have experiments going on here. I've done a lot of work at this little reef here. And then a lot of what I'll talk about today actually was done right about here. But I've worked all over this island and I've done a lot of different things here. So if you're interested in the types of work to go on here, and we have this long-term ecological research program there, like Anita mentioned, where we've been kind of going back and monitoring this, the same things all around the island for about 25 years. Not me personally, but I've been working there about 5 years. But we have these long-term records of how the island has been changing over time, which is really important when thinking about how ecosystems will change under climate change. So anyway, it's a great place to work. It's beautiful, but also it's got this really kind of interesting history in terms of disturbances. So again, disturbances are anything that can like kill coral you can think of. So in this case, in Moorea, dating all the way back to about 2005 when they started taking data out there. This graph, it's fairly simple to read, but I'll walk you through it. You have just year on the x-axis here, and then percent cover is really just, it's basically like how much of something is there on the reef. And these dots here are coral. And these dots down here are macroalgae. I would try to say what colors they are. I'm actually colorblind, so I don't want to mess it up, but I think you can probably tell for yourself. Basically, throughout the years, we've seen coral go up and down and up and down. And same thing with that macroalgae. And it's been in response to these different types of disturbances that have happened. This little spiky guy here is one of those crown of thorns sea stars. And those are those things that eat coral. They put their stomachs out over a coral and just dissolve it and kind of slurp it up. And sometimes their numbers get so abundant that they actually can just completely wipe out a whole reef. And that's what we saw here. So you can see kind of right when that happened, coral went from, around 40% cover, meaning like if you were to look down at the reef, 40% of it was covered in coral, which is pretty good. That's pretty healthy. But then it just tanked after that happened. And then right after that, we had this big cyclone event. So that came through and sort of just wiped out everything that was left. And sort of while that was happening, we had a little bit of the macroalgae coming up. But then coral were able to recover. We saw this really remarkable recovery of the corals on the reef, where they got back kind of where they were before all that disturbance happened within about 5 years. And then they shot all the way up here to where in some places the reef was covered maybe 75% in coral. It was really unbelievable to look at. Almost every square inch of the reef was just coral. But then we had another disturbance happen. Back in 2019, we had this big coral bleaching event where the water got too hot and it killed basically almost all the coral that was on the reef. It went from about 70 to 80% coral cover down to about less than 20. And you can see that it actually has been declining even since then. But in response to that, all that macroalgae kind of ticked up. So that's sort of a cool history that Moorea has had, just in terms of like how all these different disturbances that it sort of has been able to recover from historically. But we're seeing sort of a different thing happening now with this coral bleaching event. So I wanted to dig into this a little bit more. I wanted to look at, do these dead coral skeletons actually affect the ability of the living corals to grow and survive? So after this big coral bleaching event, this is what the reef looked like in 2019. I decided to set up an experiment where I went through and I actually removed all the dead coral skeletons off of like some places on the reef using these really sophisticated tools, a hammer and chisel. And so you can see this giant pile of dead coral skeletons here that I took out of just a pretty small area, just two of these plots that are 4 square meters, 2 meters on each side. So then what I did is I've, over the last 4 years, been tracking the recovery of the plots where I removed the dead coral and comparing that with places where the dead coral is still there. I'm using a technique called photogrammetry. All that is sort of a fancy word for using photographs to make measurements. And so what I'll do is I have one of these plots. So again, this is about 2 meters on each side, just a little square on the reef. I set up these special structures that help with the photo taking process. And then I'll actually take several hundred photos of this little area here that are all overlapping each other, just very slightly different angles. And what that allows me to do is put all the photos together into one giant super photo, which you can then turn into a 3-dimensional model, which maybe that sounds like very abstract, but I'll sort of show you what I mean. Okay, so if we look at this is one of my plots. It's, again, about 4 square meters. This might look like one single photo, but this is actually about 250 different photographs, all stitched together into what we call a photo mosaic. So all these, I basically swam back and forth across this, just taking photo after photo. Every kind of like second, I would take a photo and just swim really slowly back and forth, and I'd go the other way. I'd swim back and forth, do the same thing this way. And then I would swim around the entire perimeter of the plot and take photos kind of at an angle. And so again, why that is important and what that allows us to do, I'm actually going to switch over and try to show you a video. Hopefully this works. Okay. So again, here's one of these plots. Pretty sure it's the same one that I just showed you. Sort of looks like a photo, but you can actually turn this on its side. You can spin it around. You can see all the structures and the contours of all the different corals. And so this is, again, taking hundreds of photos and putting them all together to sort of reconstruct the 3-dimensionality of that whole chunk of reef. So here, this shows where every single photograph was taken from. Every one of those little squares is a different photo. And so you kind of saw those special target structures. What that allows me to do is I can actually measure the distances between all of those and create this network. It's kind of like a ruler. It allows me to make measurements inside that are really, really accurate. And so this whole model is built on what's called a point cloud. These are all just a bunch of different points in three-dimensional space. And that's how it knows basically how, some parts of the plot are higher, some parts are lower, and how everything's kind of spread out. So then underneath that is what's called a digital elevation model. This is sort of, you can almost think of this as like a topographic map. This is showing all the high points and the low points and how different they are from each other. And finally, we get back to what's called the photo mosaic. And you can see this is basically like one giant super photo. And so you can zoom in as far as you want. It's very high resolution, but it also has all that three-dimensional information in it. Okay. I see there's some things in the chat. Maybe I'll come back and I'll answer these questions a little bit later towards the end. But yeah, go ahead and keep putting them into the chat if you want to. Okay, so here's one of these big super photos, right? So it's one thing to be able to make the super photo, but you have to actually be able to do something with it. So I wanted to basically compare the recovery rates of these plots where I actually went through with the hammer and chisel and removed all the dead corals. So you see all these big spaces. All those spaces, there used to be a big dead coral there, and I took it out. And you can kind of see the difference when you look at a plot right next to it where I didn't take the dead coral out. So like all these big brown fuzzy looking corals, those are all actually dead. They died in the bleaching event. So you can see it's actually a pretty different environment for those corals that are still alive. And so I was really interested in whether or not it affects the growth of the living corals to have the dead stuff around. So the way I actually measured it is using this this program I've been helping some people develop that uses artificial intelligence to help you actually go through and measure these things. So say I want to go and measure this coral. The way people used to have to do this is you would take a computer program and by hand with a mouse, you would have to sit there and trace it out by hand. You can imagine how long that would take for every single coral, and you have to do it for a while. And sometimes you actually let your finger off the mouse and then you lose it. You have to start over. Anyway, it's a really long, labor-intensive process. So they actually have created this program that uses AI so that all you have to do is you can see these crosshairs here. You click the 4 points kind of like around the coral, the one that's furthest to the left. That's the one you can see right here. There's a little red X up here. That's the one that's furthest to the top. Another one over here. Right here. Furthest to the right. And then the one that's furthest to the bottom. You just click 4 points real quick around the coral, and then it automatically will just outline it for you. A perfect, nearly perfect outline all the way around the coral with all these jagged edges and everything. And then you basically tell it like, okay, that coral is a Poecilopora. That's like the genus name. And then it will actually measure it for you. It'll tell you exactly what the area is in 3 dimensions of that coral. So then what's cool is if I keep taking these photographs over time, I can see how these individual corals are changing. So this coral here, or both these corals here, after 3 years, they grew by like quite a lot. They almost doubled in size. But in other cases, you have a lot of corals that actually didn't survive, right? Like this big one here actually shrunk by quite a lot. This one down here died completely. It kind of turned into this brown fuzzy coral that got covered in algae. So here I'm going to show you once again, this is when I started the experiment. I wanted to show you how these two plots changed over time. So now focus your attention on the left, the one where it says structure removing. I don't know if anything changed. Here we go, 3 years later. So you can see that lots of those corals grew, but some of them died. Here, I'll do that again, actually. So you can see that, again, several of them got a lot bigger, but a few of them died. But now if you look at the one on the right, this is the one that says structure retaining. This is the way it looked at the beginning of the experiment. And then 3 years later, a lot more of that coral died. There's kind of not that much left. So kind of like what we've been seeing is that when you leave the dead coral on the reef, it's actually really bad for the rest of the corals that are still alive. They basically end up either shrinking or dying a lot more when they're surrounded and like kind of packed in by that dead coral. So that kind of makes you wonder, like, maybe like, why is that happening, right? We know that it is happening, but we don't really have an understanding of what we call the mechanism underneath it. So that's why I wanted to take the next step to actually look at how the dead coral is affecting this really important process on coral reefs, which is herbivory. Maybe you've heard of herbivory before. It's basically the process of animals eating plants, right? And in coral reefs, what that looks like is fish, in this case, in this picture here, this is a parrotfish, is eating algae. So I wanted to take sort of a closer look at this because it's really important to understand how this process of herbivory is affected, because herbivores help remove algae off the reef. And that's really beneficial to corals because coral and macroalgae compete for space on the reef, as well as light, because both algae and corals depend on photosynthesis to gain their energy. And so when you actually, so the winner of this competition determines whether the ecosystem will be sort of like a healthy coral reef like you see on the left here, or an algae dominated reef like you see on the right here. And so when you don't have enough herbivores, or the herbivores aren't able to do their job of removing algae off the reef, we have been seeing lots of cases where those reefs actually transition to being dominated by algae, which is a lot worse for the ecosystem. A lot fewer fish are able to live in algae and be supported by algae. The reefs are just much worse to look at. The water quality gets worse. Tourists don't want to dive on reefs that are covered in algae. So there's a lot of reasons why we don't want an algae dominated reef. So I did this sort of experiment where I wanted to look at how do these dead coral skeletons affect the ability for herbivores to actually get in and remove the algae. So I have these things that are called herbivory assays. It's basically just like a piece of metal fencing mesh that I attach these little bits of macroalgae to. So this is called Amansia. This is a type of algae that can grow really fast and compete with corals. And so I kind of did these paired assays where I put out one tray that had just the algae, and then I put right next to that a tray that had all that same kind of algae, but it was kind of stuck in between these dead coral skeletons. And so I recorded after a few days in each case how much of the algae got eaten, but I also took videos. So I actually got a couple more videos to show here. So here's two of these assays right next to each other on the reef. You can see that open one on the left here, and on the right, that's the one that has the dead coral skeletons with the algae in between. So here's just a quick clip of some of the herbivores in action. Oh no, it got slow again. 1 second, let me see if I can make that go a little bit smoother. So you can see Well, it's a little slow still, but trust that that's just a normal video. These fish swimming into frame, and they sort of peck at the algae or even rip pieces of it off. But you can see how there's these two fish. This is a unicorn fish and a parrotfish, both really important herbivores on the reef. And they're only going for the algae that's open. They're basically completely ignoring the algae that is in between the dead skeletons. Maybe it's just harder to see it. Maybe it's just harder to get to because they don't want to scrape their faces on those dead skeletons. We're not really sure why, but we're definitely seeing that they prefer to eat the algae when it's out in the open compared to when it's blocked by these dead skeletons. And so I have one other video. If it's too grainy or if it's too clunky, maybe I won't show it, but just try this here. This is me and my intern out in Moorea. We're just kind of setting up these cameras to film these assays. But when you set up 4 cameras once a day for 5 days in a row for 7 weeks in a row, you end up seeing a lot of different stuff you don't really expect. Like this sea turtle that came in and tried to take a bite, which I don't know, maybe they're important herbivores. I don't think people look a whole lot into that. Or in this case, we had a big black-tip reef shark swim by. Definitely not an herbivore, but still cool to see. And my favorite, the spotted eagle rays. And you can see in this case, there was sort of a whole flock of them. Once again, not herbivores, but still really cool to see them. Okay, at least that one worked. Okay, so we put out these assays. I kind of showed you in that clunky video that wasn't loading very well that the fish seem to favor these more open plots on the left because it was just easier to get to the algae. And we really saw that in the results. So this is showing one of these paired assays right next to each other. This is after just 3 days. The one that was totally open, you can barely even see the little nubs of the algae that's left over. Right next to it, though, these dead skeletons almost completely protected all that algae in between from getting eaten at all. And so we're seeing this really strong pattern where the dead skeletons really reduce the ability of those herbivores to actually remove the macroalgae. Okay, so I'll try to talk fairly quickly about the next little bit so we can kind of get to being able to just have some Q&A. But I'll just talk really quickly about this other kind of work that I do, which is actually using math to model or create models of how the reef system works so that you can test out different ideas and see how the reef might recover under different conditions. Now that we know that these things take place in nature where the dead skeletons kind of reduce the growth of live corals, and they also reduce the ability of herbivores to do their jobs and remove that macroalgae. Now you can actually use that information to make a model of that ecosystem and see how things would play out over much longer timescales. So I built a model out of just sort of a system of equations, but it's basically like 5 equations that all talk to each other. But to make it really simple, I modeled how live coral and macroalgae compete with each other for this empty reef space to grow on. But I made a couple of modifications to this. I also included an equation that captures what dead coral does on the reef and the role that it plays. And then that dead coral can actually have either living coral or macroalgae grow on top of it. And so that's important for a couple of reasons that I'll get into in a second. Whether the coral is growing on top of these dead skeletons or kind of out by itself, and whether the macroalgae is growing on the dead skeletons or out by itself. So with this model, I was able to actually simulate a tropical storm. So you can imagine a tropical storm comes through and it basically just rips everything off the reef. So it makes everything that's on the reef, all these different things, go right back to empty space. On the other hand, you have that coral bleaching type of disturbance. And what this does is it only affects the live coral, causes the live coral to die and just become this kind of dead coral on the reef, which again can then be colonized or grown on by more coral or by algae. The important thing about these corals landing on the dead coral is that that dead coral will eventually break apart. It's not a good place for living corals. What's important about the macroalgae and where they grow, like I showed you before, is that the algae that grow on the dead corals are protected from those important herbivores. So they actually have sort of a refuge by growing inside the branches of those dead coral skeletons. So I use this model and I did a lot of stuff with it. The only thing I want to show you from it is sort of how you can forecast or model the trajectory of recovery over time. So here is, I'll just orient you to what these graphs are showing real quick. On the bottom, you can think of this as just time. So this is actually years, but this is how these different things, either the coral, that top line, the live coral, the top line, dead coral, that middle line, and the algae are all changing. So if we start over here on the left side of the graph, we have what you consider a healthy coral reef. Lots and lots of live coral, a little bit of dead coral, and no macroalgae. So right at this point where that dashed line is, I simulated a tropical storm that killed about half of the coral that was there. But what you see here is that the coral kind of bounced right back, right? They have this big dip, but then right after that, they come right back up and we're back to a healthy coral reef. And we actually saw that happen in Moorea, right? Remember that first graph I showed you? The coral kind of got totally knocked out by that cyclone, but then they sort of bounced back. If we then look at a coral bleaching scenario, sorry, I keep showing this icon, I haven't really explained it. I can't think of a better way to like visualize coral bleaching. So I have a little coral in a frying pan to show that they're getting heated up. Anyway, if we start with that same reef where you have lots of live coral, a little bit of dead coral, a little bit of macroalgae, but we hit them with a coral bleaching event where, remember, all that dead skeleton gets left on the reef. This black line shoots up right when the coral get hit. But then what happens is that line down here that was kind of down at zero, the macroalgae, right after that, they continue to grow because they have all this dead skeleton to grow on and be protected from the herbivores. So the macroalgae actually take over and they become the dominant organism on the reef, and it stays that way for, in this case, like 1,500 years, which is not very realistic, but we are sort of seeing at least this part of everything unfold where the macroalgae are becoming sort of the dominant organism on the reef. So this is just one very small snippet of what you can do with a model. You can kind of look at how things are going to play out over hundreds or thousands of years if you're interested in that, and you can kind of simulate these different situations and scenarios to see how you can get different outcomes. So what we're showing here is that you can have these two different types of disturbances, and whether they leave the dead corals in place on the reef or remove them can completely change the outcome of recovery. Okay, oh, sorry, shift to a macroalgae. So a couple of takeaways from this. This dead coral skeleton, which is a type of material legacy, may slow or even prevent the recovery of corals on reefs. And this material legacy also increases the chance that the reef will shift to being dominated by algae, which is sort of not a very favorable outcome. And actually, the types of disturbances that create these material legacies, like coral bleaching or these predator outbreaks, they're happening more often and they're happening with a greater intensity. So something we might want to consider is, how can we actually, should we maybe like remove these dead skeletons off the reef to enhance the chances that the reef will recover? This hasn't really been done on coral reefs. I'm, to my knowledge, one of the first people to like try to remove the skeletons to stimulate recovery. But what's sort of promising is that in other ecosystems where you have these material legacies, people have already been doing a similar type of idea, depending on what role the material legacy plays. So in forests, when you have a big drought or an outbreak of what we call bark beetles, that will kill a whole forest, or lots and lots of trees, at least in a forest. And you have all these dead trees that are standing there. That actually makes the forest very vulnerable to fires, right? If you have a lot of dead wood just kind of standing there, it'll just go up like a matchbox if it's given the chance. So what people will actually do is they go and remove those dead trees to reduce the risk of fire, or at least the intensity of fire that takes place there. In a totally different system, people think about how they can use oyster shells to actually stimulate the recovery of an oyster reef. So an oyster reef is sort of like a whole field or bed of oysters that are just sitting in the mud. If they weren't there, there's no structure, right? It's just a bunch of mud and silt. So on these reefs, or on these oyster reefs that have had all their oysters removed, like after maybe somebody dredged them to create a harbor or something, people are actually adding oyster shells in to regain the structure and give a place for new oysters to come in and settle and sort of revive the oyster reef. So this idea of manipulating material legacies for the purposes of management and restoration has sort of already been put into place in these other systems. So I'm sort of saying, why don't we try this on coral reefs on sort of a larger scale to see if we can actually enhance the recovery of those of that ecosystem. So anyway, that's all I have for you. Thanks for listening to me, and I'm happy to take as many questions as you have now.
Well, thank you for your talk. I really appreciate it. I definitely learned a lot. I guess like before we go with questions, for those of you who did not hear me at the beginning, if you guys are here for Chloe's stats class, please send me a direct message on the chat with your name and your section so I could give that information to Chloe, please. And I actually do have, maybe we could go with the questions that were on the chat first. I know I do have some questions for you, but maybe we could go with the ones in the chat first. Let me see if I can find them.
Yeah, I have them here.
Okay.
So real quick, I'll just scroll through these questions in the chat. What is material legacy? Someone probably said that before I explained it. Because I probably said it a bunch of times before I explained it, but hopefully that's answered at this point. I'm happy to redefine that if anybody wants. Let's see. If the coral died because of bleaching, wouldn't that also contribute to them not being able to make a comeback? Yeah, so that's kind of, hopefully that's what I was showing you later on, is that like, because the corals died in the bleaching event, it's sort of not a really good situation for the corals to be able to recover afterwards. Have I heard of coral gardeners? Yeah, I've interacted with them quite a bit. They actually work in the same bay and this on the same island as me. So we've interacted with them a lot. They have some interesting, cool work going on. And our research group is starting to work a lot more with them and like try to bring a little more like rigorous science into their restoration practice, which has like gotten this huge global attention at this point. So yeah.
So there are some questions asking what kind of statistical analysis did you use?
Let's see. For the modeling, it's sort of like you set the model up with these different types of conditions. So like say you can have different starting conditions, different amounts of live coral to start, different amounts of macroalgae to start. And then you can also hit those different starting conditions with different types or different intensities of disturbance. And you look at all those different conditions and see where the system ends up at what you call equilibrium. So once you like disturb the system, it kind of goes haywire for a little bit, and then it will reach some kind of stable equilibrium point. And then that's sort of your recovery outcome in response to something like the disturbance. As far as the experiments statistically, I'm looking more at like, I guess one approach I'm taking is like, okay, it's one thing to look at for an entire plot if it has dead coral, or if it doesn't, did it recover better? But I actually want to look specifically at like each individual coral and how much dead coral is surrounding it, and quantifying the amount of like shared border it has, live coral has with dead coral, and seeing how that affects its chance of surviving and growing. So there's just a couple examples of the statistics I'm doing.
Okay. There is a question that says, what kind of camera / underwater casings did you use, and did you stick with the same kind of cameras, or did you at one point adjust the photos over time for the photogrammetry?
Yeah, so I've used a lot of different cameras to do the photogrammetry work. The stuff I showed you was just like kind of it's called an Olympus Tough. It's just sort of a digital point and shoot camera that's waterproof, and you can get a housing for it, like a plastic housing, and then something that's called a dome port lens, just kind of almost like a fisheye lens that you put onto it. Those are like fairly cheap, like relatively speaking, to get and use. I've also used some more expensive cameras that kind of freak me out to use because they're so expensive. I'm always worried I'm going to break them. But those are like Lumix DSLR cameras with these big fancy underwater housings that you put on them. And then in terms of changing the photos, and I'm trying to remember the question, in terms of adjusting the photos over time, you do have to adjust the color on the photos, because when you take photos underwater, everything basically looks blue and you lose all the red in the photo. So you have to adjust the color, which, as I mentioned, I'm colorblind. That's probably the hardest thing for me to do in all the work that I do, is correct the color of photos when I can't really even see color that well to begin with.
So I'm assuming you had to have someone to help you making
Usually
sure
I have somebody there to help into making sure I'm not making the whole photo look yellow or something.
So it's another question asking, are you still researching this? And if so, what's the likelihood that you'll be removing the dead coral?
So I have like that first experiment that I did where I removed a lot of dead coral is still kind of ongoing. I think I probably have taken the last set of photos that I'll do at least while I'm in grad school still. There is a lot of interest, at least in my research group, to do this on a larger scale now. I told you the plots that I'm working with are 4 square meters, so they're really not that big. We're talking about doing it over like a much larger area to see if we can like get these big, large scale responses for recovery.
Okay.
And there are a couple other questions I saw in the chat. There's one, are coral able to recover from bleaching? Is it reversible? Yes, in some cases. It depends on how bad the bleaching is. The thing is, corals are usually very resilient. They're usually able to recover very well, but bleaching is this sort of new type of thing that they're having to experience. They're having to deal with it. When a coral is bleached, it's actually not dead. It's sort of an intermediate zombie phase where they're not really alive, they're not really dead. They can actually recover from it sometimes as long as the water cools down quickly enough.
Okay.
Which actually is a response to that says exactly that. Yeah.
Okay. So I did see a hand raised. I don't know if he's still here. Connor.
Mm-hmm. I typed the question in the chat, but I'm currently an undergrad. I'm a marine bio major, and I really want to get my scientific diving certification. So I'm trying to find like how I can get that as soon as possible, because like my local institution that's through my school has like they're not doing it until June last I checked. So
okay.
But I really want to get started with that.
What year are you in your program?
I just transferred. I'm a junior.
Okay. And what university are you at?
Cal State Long Beach.
Cool. As far as I know, they have a good program. There's some researchers at Cal State Long Beach that actually work in Moorea that we see sometimes.
Oh, awesome.
If you get your scientific diving certification at any point while you're in college, that's a good thing. And if you get it in your junior year, that's pretty much like, I would say, pretty standard. It's pretty rare that people get it before that. So I think you're totally set as long as you can try to get into that class then. That way you can kind of get set up to help out with some research diving in your senior year, or maybe the summer after your senior year.
Right. That's what I'm aiming for. But that's what I was kind of wondering, is that locally there's this one place, but they don't offer it until June. So I was like wondering, are there other places around Southern California? I'm not sure where you live, but if you are, then that offer it year round, like certifications. Yeah,
it's sort of like most like universities that have a marine biology program or like a diving program at all have an AAUS class. Like ours here is pretty impacted, so it's always full, and it's pretty tough for even like the Santa Barbara students to get in. But you can look into other programs, like there's Santa Barbara City College that I think does a science diving class, and you can see if they have full classes. And I would say at most they usually do probably 2 classes a year because they're pretty intense classes to teach.
Mm-hmm. Right. Thank you very much. I really appreciate the information.
Yeah, of course.
So there is another question asking, how long did it take to stitch the photos together for the photo mosaic?
That's a great question. Depends on how big of an area you're covering and how many photos. So like those small ones don't take that long, actually. Probably take like 10 minutes on a really high-speed computer. It's different when we do some bigger plots where we're covering a much larger area, like maybe like 150 to 200 square meters. And we take probably like 1,000 to 2,000 photos of those areas. And so when the computer has to deal with like figuring out how to stitch that many photos together, it can take more like hours, I would say, but not much more than that.
Okay. There's someone else who was saying, would you say that it is a poison disturbance for the experiments?
What do you mean by that? I know what a Poisson distribution is, but like, what do you mean for the experiment?
Yeah, it would be like a Poisson distribution. So were you using that as kind of like a way for your experimental analysis portion? Because I know you use the plots and you're kind of observing them over time. So would you consider that as a Poisson distribution?
No, I don't. I don't know exactly. Yeah, I guess I don't know exactly what you mean by that. Like a Poisson distribution, as far as I know, is like a statistical situation for data where you have most of your data are kind of skewed towards one side. But as far as the experimental design, I don't know if that really comes into play there. If it comes, once it comes to the statistical analysis, like a Poisson distribution could be something like, if all the corals died, if most of the corals died and there were a few that survived, I might end up with a Poisson distribution, because all the corals, all the ones that are dead are all to one side of the, the graph or the relationship.
Gotcha.
Yeah.
No, that helps. Yeah, thank you.
So there's another question that's asking, do you have a final outcome for your research already? Or are you still working on teaching it? And what is your ideal outcome from your overall research?
Yeah, that's a good point. Like, why am I doing all this? I think the answer to that is that I mean, first of all, this is what I've been doing for my PhD research. So hopefully within the next 8 months, I'll have a PhD. And that will be what I did to obtain it. But again, like, I really am interested in science that has some sort of application or purpose, especially related to conservation or restoration. So I think the idea at this point is to try and at least build enough evidence that this like dead coral skeleton material legacy idea is something that people who are like trying to conserve coral reefs should really be thinking about, in terms of like, how do we best stimulate recovery and enhance resilience of the coral reefs that we have that are facing all these threats in the modern day.
Well, I think those are all the questions on the chat, I believe. Wait, oh wait, no, there's one more. What is the process called that you pick a section of the coral reef called?
Sorry, say that again.
What is the process called that you pick a section of the coral reef? Oh, I think it's when you were removing the coral reef from like the dead coral reefs.
Yeah, so when I like select plots, or like site selection, that's sort of like, when I was doing that, I tried to get as many plots that were like very similar to each other. So I would like kind of go over the reef, and I was basically drawing squares with these, we call them transect tapes. It's like a long measuring tape. I was just basically making squares that like encompassed a similar amount of live and dead coral to every other square that I was making. So I wanted all my plots to be really similar in terms of how much live coral and dead coral were in them.
Okay, well, now that we have those questions down, I do have a couple questions for you.
Okay.
So the model that you use where you be able to put all of the pictures in the mosaic area, does the model know like where each picture goes, or do you kind of have to? Okay.
Yeah, it knows exactly where each picture goes.
Okay. How often did you remove the coral? Because I know you said you removed the coral at the beginning, but did you like remove the bleaching corals at the beginning and just left it like that? Or like, would you go back and remove some of it every once in a while, or just leave it?
I probably should have done that, but I actually just did it once. I just did it at the very beginning because I thought like, okay, that's what like a disturbance is. It's what, like one event that happens at one point in time, and I wanted to just try to resemble that as best I could. So like a storm that came through and just like killed coral like all in one fell swoop, I wanted to just do it then and then see what happened afterwards. So I only removed coral one time.
Okay. Some of the models that you were talking about, is that a model that I might have missed it, but is it a model that you created, or was it a model that someone else created and you were using that model?
So you're talking about the math model or the?
Yes.
Yeah, okay. That one started with a model that someone else had built, and then I modified it. So someone came up with a whole set of equations to like model, coral and algae and empty space. And then I added a few equations onto it to say like, okay, I want to think about dead coral as well, and algae that grows on dead coral, and live coral that grows on dead coral, and all that stuff.
I guess there's a couple of questions that came in. What is the name of the AI that you've worked on creating? Is it public?
Yeah, it is public. It's just not very like fully developed yet. It's not like commercial yet because we're sort of still working on it, but you can download it and like work on it. It's a program called Tag Lab. And it's basically, it takes these these photo mosaics, you put it into it, and then you can just go through and kind of say like, okay, I want you to draw a border around, this object with, in between these 4 points. We've actually gotten it to the point, we just put out a paper recently where you don't even have to click any points anymore around the corals. You just put a big photo mosaic in with like 1,000 corals in it, and you say, you find them all. And then it'll go through and it'll outline every single coral for you in like 10 minutes, which is, a lot better than me going through. And even with the 4 clicks on each coral, that takes me hours.
Okay, so those were the only questions I have. I don't see any other questions in the chat. If there's any other questions.
Sorry, I had a quick question. So the removal from the first time you took the coral, that was between the research the research you gathered was that was in the span of 3 years, right?
Yeah, what I showed you was 3 years, and I think those results were like about a year ago. So I just got back from the island, and I took another set of photos for each plot. So I now have 4 years worth of data and information, but I haven't gotten through processing all that yet. But sorry, maybe you have more to your question there.
Okay, thank you.
Oh yeah, totally.
Is there any other questions for Kai? Okay, well, if there isn't any more questions, thank you very much for coming. I definitely enjoyed listening to your talk. I definitely learned a lot more about coral reefs, more than I did before. And it was really great to see how your research is helping get more knowledge on corals and how to help them in the future. So yeah, thank you so very much for coming. I hope you guys got to learn more about coral reefs, and hopefully you guys could be able to attend our next talk next month, which is going to be focused on the bumblebees. So yeah, and if you guys haven't had a chance, again, if you guys are in Chloe's class, just send me a direct message and then just let with your name and your section, okay? Thank you, you guys.
Yeah, thanks everybody.
Thank you so much for the information.
Yeah, of course. My pleasure.